everyone's attention? Okay, awesome. Good afternoon and welcome to the UCSF Academic Senate 68th Annual Faculty Research Lecture in Basic Science. I'm Melissa Medved. I'm an Associate Professor of Pediatrics and Chair of the UCSF Academic Senate Committee on Research. So it's my honor today to present the Faculty Research Lecture. The Academic Senate's Faculty Research Lecture started in 1957. The first lecture was given by Professor Melvin Calvin on the topic of photosynthesis as part of the Charter Day activities. And originally, an ad hoc committee was appointed to nominate the first faculty research lecturer. From 1960 on, the Committee on Research decided to take on this responsibility, and in 1966, decided to solicit new nominations each year from the deans, department chairs, institute directors, and members of the Senate. Since then, nominations are made by UCSF faculty who consider scientific research contributions of their colleagues and submit nominations for this prestigious award to the Senate's Committee on Research. So I'd like to thank the committee for their hard work selecting this year's winner. This Faculty Research Lecture Award recognizes exceptional achievements in basic science research, and today we're proud to honor our colleague, Dr. Jason Cyster. And now I'm pleased to introduce Professor Mark Anderson and Professor Mark Ansel, who are the nominators of Dr. Cyster. Dr. Anderson's a professor of medicine, director of the UCSF Diabetes Center, the A.W. and Mary Margaret Clausen Distinguished Professor of Metabolism and Endocrinology, and the Robert and Michelle Friend Endowed Chair in Diabetes Research. Dr. Ansel is a professor in the Department of Microbiology and Immunology and focuses on immunology, cancer biology, and cell signaling. So pass it over to you guys.
Hey, thank you. So the two Marks want to make just a couple of quick remarks here. First, obviously, I want to thank the Academic Senate and the Selection Committee for making a fantastic choice. My colleague Jason Cyster, and Jason, congratulations. I just wanted to say a few words on behalf of the perspective of the faculty in the immunology program here. We're in for a real treat today, but Jason is really just been a special person in my life as an investigator at UCSF. And you can see all the people that he's mentored have made the trip here to be with us today, but he's really brought out the best in all of us. And, just a quick anecdote in the lab is when, we run into a roadblock in the lab and things get difficult, a not uncommon thing that happens is somebody will say, "Did you talk to Jason?" See what he thinks, right? You may not have the answer, but he'll have a thoughtful suggestion. And so, Jason, I just want to thank you again for bringing out the best in our science and the bar for science. And it's just been such a treat and pleasure to have you as a colleague. So congratulations. And now I'll hand it over to the other Mark.
Yeah, this is an honor just to get to stand up here and introduce Jason. Thank you, Mark, Kevin, Anita, Julie Zickerman, and others who made this possible today. Thanks to the Academic Senate. And I have a little bit more to say about Jason. There's another one. So I've known Jason for quite a while. I'm his neighbor up on the 10th floor, and I was also a graduate student in his lab from 1996 to 2001. Some of you might know that Jason grew up in rural Australia on a farm, some hours away from Perth, as pictured here. And he, like me, was really inspired by David Attenborough, the great naturalist of BBC fame during that time. And this quote from Attenborough, I think, could apply equally to Jason as it does to its originator. "I know of no pleasure deeper than that which comes from contemplating the natural world and trying to understand it." And you can see that in Jason's dedication to biological research all the time that we've known him here at UCSF. Jason took that inspiration and curiosity to the University of Western Australia, not too far away at first, where he did his bachelor's degree, and then on to Oxford, where he did his PhD in immunology with Alan Williams, and then to the Bay Area, where he had an extraordinarily productive and efficient postdoctoral training period with Chris Goodnow down at Stanford University before arriving here in 1995 as an assistant professor. And it was during that time that Jason made some really seminal observations about the behavior of B lymphocytes, so immune cells that can make antibodies, control humoral immune responses, and the change in the patterns of their homing and localization within secondary lymphoid organs, so like the lymph nodes and the spleen. And these are some pictures he'll probably show again, or similar ones from that time. And see that the B cells usually hang out together with their brethren B cells, they're the red ones, in these things that we call follicles. And after they're called into an immune response, when they find something specific they might be able to make an antibody to, they relocalize, move out of the follicles, get excluded from the follicles, and line up in these adjacent areas, often close to where the T cells live in the cortical areas. And the interactions that happen here are very important. So I met Jason around this time. It was in February 1996, and I was an aspiring interviewee for the biomedical sciences graduate program. And Jason had either this or a picture a lot like it tacked to his wall. And he explained it to me and told me, show me this really striking result and what it meant. And I was like, an ambitious interviewee trying to say something smart. And I think I said something like, wow. and Jason, with maybe a hint of a wry smile, he said, yeah, so you have a good afternoon in the lab and then you get back to work to figure it out. And that, I didn't know it at the time, but that's so quintessential Jason. It demonstrates that driving curiosity, the relentless work ethic, and also the humility that he brings to his science. And these are traits that Jason has not wavered from throughout the 30 years that ensued, and going strong, continuing today. And there have been some major contributions. So his lab, Jason, have visualized with ever improving technology and with ever greater precision and elegance the movements of B cells and other cells into the lymph nodes and other lymphoid organs, all around within them, how they interact, and also importantly, how they leave. He's identified the molecular basis of this dance, the choreography, the cues that move the cells around and make them interact with each other and find different positions. And importantly, he figured out why it matters. And these movements and interactions are extremely important for the development of both the initiation of immune response, the maturation of immune responses, and also for immune tolerance, this characteristic of our immune systems where they're resistant to making a big response against self-antigens, parts of our own body. Along the way, he's made additional fundamental discoveries about the development of lymphoid organs, the tertiary lymphoid structures that are like the lymphoid organs that appear in autoimmunity and inflammation and cancer, the signaling and regulation of G protein coupled receptors. He doesn't want me to take up too much of his time or steal too much of his thunder. It goes on and on. And Jason's accomplishments have been recognized with an equally crazy long list of accolades and awards. Some of them he's been inducted into the National Academy of Science, the American Academy of Arts and Sciences, he's a recipient of the Harrisonburg Award, and also a distinguished fellow of the American Association of Immunologists. This work is chronicled in over 270 papers with over 50,000 citations. So his work is read widely, appreciated, and very influential in the field. As Mark alluded to, Jason's impact and his importance to the UCSF community and the immunology communities goes beyond these great scientific research contributions. And really, Jason's an exemplar of something that I really cherish about this institution, and that's that many of our very best scientists are also our best community members, servants, leaders, educators. When it was needed, Jason stepped up to become the director of the biomedical sciences graduate program, and together with Lisa McGargle, they doubled the size of the program while simultaneously improving the quality of the students that came in and also the quality of the education that they received here at UCSF. Jason held that role for 11 years, an eternity in graduate program directorship. and very soon after stepping down, he became the director of the immunology training program, which is something he still is today. He's also a great mentor, I can tell you personally. But if you really want to see the depth of Jason's impact on the field and also the depth of appreciation for the people, the dozens of postdocs and students and postbacks and other trainees that have worked with him over the years, You can come to the same room tomorrow, starting a little before 9, finishing a little after 5. We're going to have a symposium. The architecture of immunity is almost entirely populated with lab alumni who have traveled here from all over the place. Can I get all of the current and all of the former Cyster Lab members to raise your hand? Right. Yeah. We'll travel from Israel and Japan and Switzerland and the UK and all over the States to be part of this and to be here with Jason for this day today. Thank you guys. I'm proud to be one of you. Jason's also a great family man, whether it's helping out with the farm back in Australia, which he's still really involved with, or with Lucy raising city kids, Tanya and Tyler here in San Francisco. These pictures are from the farm and they're kind of they're basically grown now. But Jason made time to be really good at this too, and I know he takes great pride and joy in their creative activities as well. But ultimately, appropriately, Jason is best known and revered really for his dedication to his craft. Rigorous, creative, precise, solid science that stands the test of time. And I'll leave you with something I learned from Jason, maybe as a mentee or maybe more by observing him. And that's that what we perceive as insight or even intuition is often just the result of careful, thorough, attentive study. So it's a great honor to introduce Jason for the 68th faculty lectureship in basic science. Thank you, Jason.
Well, thank you, Mark and Mark. It's a real pleasure. Those very generous words and just such an honor, but also humbling knowing who's gone before me in this series. but really a privilege to be able to use this time to celebrate the work of past and present trainees. So to begin, in the lab, as Mark's already begun to introduce, we study the cues guiding immune cell movements and interactions within lymphoid tissues and the barrier surfaces with the goal of understanding how correctly targeted and regulated immune responses are mounted. And so lymphoid tissues include the primary lymphoid organs where lymphocytes in particular are made, but we'll be focusing on the secondary lymphoid organs, which include some 500 lymph nodes in humans, these blue dots distributed along the lymphatic system, some 100 pairs patches along the small intestine, the adenoids and tonsils in the nasal and oral cavity, and the spleen on the blood circulation. In addition to the secondary lymphoid organs, in infection or in cancer or autoimmune disease, ectopic or tertiary lymphoid structures can arise and contribute to those situations. And what we understand about secondary lymphoid organs can inform us about these tertiary structures. But just the sheer mass and number of these tissues tells us they must be doing something important. So if we focus in on the lymph node and think about how that is functioning to support immunity, we know that lymphocytes are continually entering from the bloodstream, and then they're going to survey that organ to see if there's something there they recognize. They're coming in across these specialized high endothelial venules, HEV, and then they're spending some hours surveying. And we need to remember that in the lymphocyte compartment, B cells or T cells, only as few as 1 in a million are specific for any given antigen due to the way the repertoires are generated. So we need these lymphoid tissues to serve as hubs to bring these rare cells together with the antigen that they might recognize. But a lot of the time, they're not going to recognize anything, so they'll move on. They'll exit after some hours, return to the lymph or exit into the lymph from a lymph node, and that will carry them back to blood circulation, and they can travel to one of the many other lymphoid tissues to continue the surveillance. But if something does come in, an antigen, a pathogen that will give rise to antigen, it will enter via the afferent lymphatic, maybe something from the skin, and that will lead to material entering the lymphatic afferent lymph and reaching the lymph node, where it could then be engaged by that rare few lymphocytes to initiate an adaptive immune response. So these tissues are organizing key events for adaptive immunity. And we've known for a long time that they're organized into 2 main compartments, B cell rich follicles or adjacent T-cell rich zones if the follicle is stained in brown here. And then the high endothelial venules, the blood vessels of which the cells are continually entering, are stained in red. But while we've known this organization for a long time, and it was well recognized from when I was starting here in the mid-90s, we really knew nothing about how that organization occurred. And so that became a major interest in the lab. So the parts for the talk, I'm going to begin with some personal background, as is traditional for this lecture, and my good fortune with mentors, which I think is also, very much what is the usual for this lecture, that we're so fortunate for our mentors. And then launching the lab, where we have focus on sniffing out the scent of a follicle. And then just 5 years in, we were looking for a way out. Then we got going on intervital imaging, and that'll give me a chance to show some movies. But then I also certainly want to save time to thank a lot of people. So as you've heard, and some of you know well, I'm from Australia and grew up mostly near a small town, happened to be called Denmark, some 20 miles from that town. You saw a picture of this house from a different angle. We were back there in June with the family. We don't live in, my parents don't live in that one anymore. They've moved to a different part of the farm. But I show it because if you step out on the front sort of walkway there and turn around, then that's what you see, some part of the farm that we grew up on. Only a couple of the animals, the cows are visible there, but there's many more down near by the river. And so to give you a flavor of what my youth was like, I'm going to indulge a bit and show a family history by our daughter, Tanya, from 2017 when she was in multimedia class in grade 8. And this is a 2-minute, and I'm hoping that sound is going to come across okay.
Yeah, I would get up early, and depending on the time of the year, it might just be getting light, and the kookaburras would be laughing out in the trees nearby, and I'd walk over to the dairy a couple hundred yards away to get the milking cow in number 55. And on the way over, we had like 6, 7, 8 black cats, and they would follow me over because they knew what was coming. And the cow would come into this old dairy, and she'd be waiting there because her milk had accumulated overnight because I'd shut the calf up, her calf the night before, so that her milk would accumulate. And she's probably been uncomfortable by this point, wanting to let that milk down. And so but she also knew there was a bucket of feed waiting for her, and so she'd go and find that bucket, which was just behind the crush, and then I'd close the crush around her neck to keep her there, and then start milking into a bucket under her udder and milking away. And then all this time, the cats have sort of come up very close, keen, waiting, and a few of them would sometimes venture in and start to try and lap out of the jug, so I could flip them away, keep milking, and then the jug would fill up, and then I'd grab this old milking churn lid and milk into that, and put that over for the cats, and they would crowd around in this sort of circle of black cats and lap away at this milk. And then I'd let the cow out of the crush and let the calf out of its pen, and they would come together, and then the calf would really latch on and start drinking to get its fill. And I'd carry the jug back to the kitchen and put it on the table for breakfast.
And that was, daily activity. And occasionally, as the calf got like a year old, they grow quite fast. It would sometimes get too sort of energetic about trying to get back with its mother. And so you can end up with this situation. And so I'm trying to address the problem. And partly, that is to say that on a family farm, you actually often have to work out how to address your own problem. So one of the experiences you get from that. In addition to cattle, we were a cattle farm. They still are farming cattle. We had some sheep, chicken, some horses, but also pigs. My mother and I, my father would have nothing to do with it, but my mother and I had a large herd of pigs, to produce for market. And I think it was the pigs that really connected me with thinking about infectious disease the most, because as you know, piglets are born very small, and there can be a lot of them per litter. And often we'd lose a few to presumably infection. And so that sort of got me thinking about what was going on there. And I actually used to try and get pamphlets to learn more about that, back before the era of internet or anything. So So from there, I was fortunate to go to the University of Western Australia up in Perth, you know, a little bit different scenery, but situated on the Swan River, University of Western Australia, beautiful campus, maybe mixed in other ways, but I was fortunate in my fourth year, in the honours year of my bachelor's degree, to work with Wayne Thomas, a brilliant immunologist who had at that time cloned the first important allergen, DERP1 from house dust mite. It's actually in the feces of the dust mite that you breathe in. And he was wanting to characterize the T cell epitopes. You know, this was back in the 80s, so early days. So it was an early process to try and do, but it was an exciting challenge to get involved in. And I really appreciated how Wayne took people like me into his lab and taught us so much. His knowledge of immunology was just really deep, and his rigor was always evident. And his willingness to sort of help support the early trainee and tolerate our mistakes with good humor was all appreciated. So I'm really, you know, I just reflect that those first months one has in a lab are so important to one's career and getting passionate about a subject. And Wayne definitely fueled my passion for immunology. So I thank him for that. And from there, I got a Commonwealth Overseas Studentship and was fortunate to go to, again, quite different scenery, to Oxford, beautiful town. And I was working in not quite so scenic building, but the Sir William Dunn School of Pathology in the MRC Cellular Immunology Unit, working with, as Mark mentioned, actually another Australian, Alan Williams, and his senior colleague, Neil Barclay. And Alan at that time was one of the first to characterize T cell surface molecules using monoclonal antibodies. And he was also the sort of father of the Ig superfamily. And I worked on CD2 and CD43 on the T cell surface. And it was really great working with Alan. I was told he was a straight down the line guy, and some people found that a bit difficult, but you know, if you worked hard, you got his respect. We had a good time. But sadly, he died young. He developed lung cancer, even though he never smoked. And I was fortunate to finish up with him just before he died, but it's a big loss to immunology. But the Dunn School was also known for, in the same unit actually, before Alan took it over, the James Gowans. Some of you will know of him. He discovered the concept of lymphocyte recirculation. So he showed that the cells that went into lymph nodes, which had been recognized, that they came out into the lymph, out of lymph nodes. And so the recirculation was occurring. And I think that sort of was in the walls of the MRC unit and permeated into me as a fascination of that recirculation concept. So from there, I was fortunate to go and work with Chris Goodnow, another Australian who had just moved, you know, it was something about I'd only trust Australians, moved to Stanford to start his lab a couple of years before. And so I was really fortunate to get into his lab. And he sent me these photos a few years back. And he comments, you know, these photos are clearly staged, looks to me like run-of-the-mill development in the bone marrow on that screen. And then in this one, makes no more sense now, whatever I'm drawing on the board, than it did then. But he would say that in this, you know, Australian sarcasm. But we were actually probably having a very deep conversation where he's just providing profound insight with his incredible depth of knowledge about immunology. And he's such an inspiring mentor, and so thoughtful and creative, and just always engaging with his people in ways that get you thinking, get you excited. And it was such a pleasure and privilege to work with him and to have been supported in all these years since by Chris. And so Chris, already as a student, had focused in on a really profound problem. So immunology, as most of us know, one of the fundamental issues is how you avoid autoreactivity, autoimmune conditions. And that's this concept of self-tolerance. But a challenge in studying that with lymphocytes is that the frequency of cells specific for any given autoantigen, just like foreign antigen, is extremely rare, maybe 1 in a million. So how do you track the fate of that cell? What Chris recognized early on when the first transgenic mice were being made is that if you make an immunoglobulin transgenic mouse, you will have a monoclonal mouse where you'll know the specificity of every B cell. And he had the further realization that he could control whether the antigen it recognized was present in the animal or not, as in he could make it a neo-self antigen. So he made his immunoglobulin transgenic mice specific for hand-in-glycosyme, which was a well-studied model antigen at that time, and made transgenic mice expressing hand-in-glycosyme. And then he could cross them together and look at the tolerance processes. So I want to just introduce one project that I worked on with Chris, because it's going to become relevant to the work that we then continued on in the lab, as Mark's already alluded to. And that was to ask about the fate of B cells seeing autoantigen for the first time in the periphery, if it was a peripheral restricted autoantigen. And so this was work I did with Suzanne Hartley in Chris's lab. And we would take his transgenic, immunoglobulin transgenic B cells specific for the known antigen and transfer them into mice that either lacked it as a self-antigen or had it as a self-antigen. So if it's not present, the B cells after transfer, you can find them in the animal, in the spleen, other lymphoid tissues readily for at least the 5 days in this assay. But if the autoantigen is present, they're eliminated within 3 days. And an important additional observation here was to look at, okay, well, where are they before they're eliminated? And so looking at day 1, we could see the brown transferred B cells were intermingled with the endogenous B cells in the B cell follicles here in the spleen, as they should be. They're doing their surveillance process for a few hours, and then they'll move on and keep doing it elsewhere. But the ones that are seeing self-antigen at this 1-day time point are excluded from the follicle, what we called follicular exclusion. And we then made the inference that, as stated in the title, that competition for follicular niches excludes self-reactive B cells from the recirculating repertoire. But we recognized that this is a correlation, that the follicular exclusion is correlated with the elimination. We had not established causation. Now, the reviewers were okay with that, but I was not. And so I was on a quest from here to work out if there was causation, which meant we had to understand how does this exclusion occur, and we had to back up because we realized, as I mentioned earlier, that we actually don't understand how the cell is positioned in any of these locations. And so I began that quest while still in Chris's lab. And that was to take advantage of some initial data about G protein coupled receptors having been found to have a role in inflammation, in chemoattraction of cells to sites of inflammation. And the class of G protein coupled receptor involved coupled to G alpha I containing heterotrimeric G proteins that would trigger this directed migration to the ligand, to the chemokine. And it was well known that there was a toxin from Bordetella pertussis that can inhibit selectively G alpha I. And one can treat cells with that and then take them away from the toxin, they'll still be inhibited. And so I treated some B cells, either with saline or with the toxin, transferred them intravenously into normal mice and looked at their location in the spleen 1 day later. And we could see that the control treated cells have co-localized with the endogenous B cells that are green, the transferred ones are orange, in the B cell follicles doing their surveillance. But if they've been pre-treated with pertussis toxins, so they can't signal through chemokine receptors, they completely fail to go into the follicles and they're stuck out in the red pole. So these data implicated G alpha I coupled receptors in B cell homing to splenic follicles. So with that in hand, and Chris generously let me take that one project out of several I was doing on to my new lab. And I was fortunate to be recruited here back in 1995 and start out on HSE3 just around the corner with neighbors Nigel Killeen and Kevin Shannon. And Kevin is here today. I couldn't have asked for a better collegial set of neighbors, and certainly the humor as well was a fantastic part of that for anyone who knows Kevin. But also I want to thank one of the attractions of UCSF. It was a small immunology program back then, but there were these immunology role models that were really important to me. Art Wise, Tony DeFranco, Rich Loxley, and Steve Rosen. I know at least some of them are here, so thank you for coming today. And so we went ahead and expressed this as a recombinant protein, this EST, and measured migration of B cells and found this remarkably efficacious attraction. The best ever thing before that was this SDF1, which was actually far better than anything else at that time, but this efficacy was just remarkable. Potency not high, but efficacy off the charts. And so we named it B lymphocyte chemoattractant, BLC. And then Vu, Mark, and Erick showed that it was a ligand for BLR1, that receptor that was abundant on B cells that we'd been looking at. And Vu performed these beautiful northern blots, which are these things that tell you expression of transcripts. And you can see in the lymphoid tissue, just so selective and so strong. This is a lymphoid chemokine. And so Mark then took on the goal of knocking it out in the old school days by homologous recombination, embryonic stem cells, with a lot of help from Nigel Killeen, our neighbor, who was really an expert in that. And after a lot of work getting that knockout to work, was able to see that indeed it was the scent of a follicle, because when we look in his knockout mouse, compared to the wild type, where you can see these tightly packed brown B cell follicles near the capsule of the lymph node, there's just this mixed up zone of B cells and T cells in that region in the knockout. So this was attracting B cells into the follicle. It was the scent of a follicle. And then we needed to understand also about the T-zone attractant, and several groups contributed to that. Dee Gunn particularly, but several groups characterized what was SLC, and Vu performed in situ hybridization in a series of lymph nodes here to show its expression in the T-zone. And Vu, in his own work, identified the mouse ortholog of ELC that was also, he showed, in the T-zone. These are adjacent sections. So you can see BLC expressed by stromal cells in the dark color here in the follicle, and these two chemokines in the T-zone. As I said, this is a ligand for BLR1. Now, unfortunately, there was renaming going on, of course, nomenclature. So BLC sadly became CXCR13, but BLR1 became CXCR4, and then these ones renamed. But I highlight that they were both ligands, or are ligands, for EBI1, which became CCR7. In addition to Vu's work there, I'll just mention a couple other chemokine achievements along at that time. Lucy Tang identified mouse MDC, which became CCL22, and showed it had a role in activated CCR4 T cell migration. And Radab Matlubian, who's of course here as well, with a tip from Art Wise actually of a chemokine-related sequence that colleagues of his identified. Merdad deorphanized or found the receptor for what became CXCR16, which was a receptor called Bonzo, which sadly got renamed CXCR6. So coming back to this observation, that we've got B cells that are in follicles here in the spleen, endogenous in brown, cells that we know the specificity of in red, but there's no antigen present. We inject the antigen that the red B cells can see, and within 6 hours, they've undergone this follicular exclusion process. So we can now think, is that due to increased response to the T-zone chemokine or decreased response to the B-zone chemokine? And so this was work Karen Rafe did in the lab, and she's here today. And we could see by staining with a reagent, actually, I think Paul Hyman made, of an FC fusion of ELC, that in 6 hours of stimulation, the B cells increase. They already have a fair amount of CCR7, not as much as a T cell, but they have some, and they go up just 2 to 3-fold. Whereas the CXCR5, hopefully you can see there's two lines superimposed. The antigen had no effect in this in vivo exposure. And when you take the cells out and do a migration assay, you can see that this T-zone chemokine acting through CCR7 is much more attracting these antigen-stimulated cells compared to the control situation. So that then is CCR7 needed for the follicular exclusion process. And so Karen tested that by taking the transgenic B cells, they're either wild type or CCR7 deficient, and exposing in vivo to antigen. So this is 12 hours after exposure, but the same thing, they're follicularly excluded, the blue cells, antigen engaged, out of the follicle next to the T-zone. But if they're CCR7 deficient, they actually completely fail to do that, end up going to the opposite direction, still in the follicle, but at the back of the follicle. So now we could come back 10 years later to this question, is it correlation or causation? And so this was work that Erick Ekland did, and he's here. And that was to take the transgenic cells, inject them into mice, either lack or contain the autoantigen. This is the wild type. As before, we see within 3 days they're eliminated. The CCR7 deficient cells, which are not follicularly excluded, contrast to these, are also eliminated. So we disproved our model. So I'll partly take you through that to say that it's okay to disprove a model. You can survive and go on. But I would say that it's best to disprove your own model rather than having someone else. So what is going on? So Robin Leslie, as a graduate student in the lab, found that autoantigen binding B cells have reduced competitiveness due to increased dependence on the B cell survival factor BAFF. And I'm not going to have time to go into all that, but I will say that BAFF is not restricted to the follicle, hence the lack of connection here between exclusion and elimination. So having gone into characterizing cues involved in organizing lymphoid tissues, we realized there was a lot more we needed to do. And so coming back to Mark, Vu, Paul, Sanjeev's paper, we noted that these specialized stromal cells in the follicles called follicular dendritic cells, darkly stained here compared to the light blue on some B cells, were missing in the BOC knockouts. It wasn't just that the B cells weren't organized, the stromal cells were missing. And that connected to an observation that David Chaplin's group had made, that the TNF family cytokine lymphotoxin alpha-1 beta-2 was needed for B cell follicle formation. And this cytokine can be expressed on B cells, they'd found, and the receptor can be on various stromal cells, including potentially the FDC precursors. So this led us to think, well, maybe there was something in the same pathway here. Maybe BOC was needed for lymphotoxin expression. And this was something Vu tested and found that when he exposed follicular B cells to BOC for a short period, compared to the control cells, there was strong induction of lymphotoxin, and that occurred in a GI or pertussis toxin sensitive manner. So the model that came from this is that we had these nascent stromal cells in the follicle area of a lymphoid tissue that make a low amount of BOC, and that can start attracting a few B cells, but not just attract them, induce them to express lymphotoxin alpha-1 beta-2, which they then display on the surface and engage the lymphotoxin beta receptor on this stromal cell to start upregulating BOC in a positive feedback loop. And then you get more BOC, more B cell recruitment, and that amplified lymphotoxin, which is an NF-kappa B type signal, promotes maturation of the stromal cell into a follicular dendritic cell. So a positive feedback circuit. Now, while we were working on some of this, we considered the possibility that BOC might be sufficient to drive entire ectopic lymphoid tissue formation. And this was something that Sanjeev Luther beautifully tested in the lab. And he's here today from Switzerland. And he leveraged Doug Hanahan's work, who'd been in the diabetes center at UCSF back then. And one of Doug's major contributions back then was to discover or to develop the rat insulin promoter that could be used for transgenesis. So you could drive gene expression in the beta cells of the pancreas. So they're the beta cells in the pancreas making a lot of insulin, but in Sanjeev's RIP-BOC transgenic, they're still making insulin, but you can see they're making a boatload of BOC as well, which normally they're not doing. So what does that make happen? You see a bunch of B cells come in, which makes sense, tracks B cells, but you also see T cells organizing in a T zone, and dendritic cells and HEV. You get an organized tertiary lymphoid structure, which Sanjeev nicely diagrammed here of this BOC promoting the B cells, which then drive the downstream events in a lymphotoxin-dependent manner. Now, connected to that, Mark had observed, and some other data also, that BOC, the knockout mice, actually had a lymph node developmental defect. So it wasn't just a follicle defect. There were a lot of lymph nodes missing. And that could not be explained by the B cell recruitment step because B cells are not needed for lymph node development. And so what we and others showed is that there was a CXCR4 positive, lymphotoxin positive lymphoid tissue inducer cell type present in early development, hematopoietic cell, that is being recruited by BOC and needed for lymph node development. So I'm not going to be able to go into that, but because of that work we did in that space, I was asked in 2014 to write a Pillars of Immunology celebratory article review thing about this seminal paper from David Chaplin's group, showing that when you knock out lymphotoxin, you lose the lymph nodes entirely. And I'm going to use this as a sort of moment for an interlude to say that I think there's a lot about how we see the world as we travel through life, through the prism of our youth. And so I want to give you a sense of why I came up with that term at the beginning of this title by showing another family history by our son in the same class a few years later.
So when I was about your age, back on the farm, my dad and I used to go and blow out stumps and dead trees for neighbors because they needed to clear their fields so that they could be better for making hay and for animal feeding. And so we would load up the ute with explosives and drills, equipment we'd need, drive over to the property, and the farmer would show us the field where the dead trees and stumps were, and get our equipment out and start drilling a hole under this tree, sort of going in at like a 70-degree angle, initially starting with like a 3-foot long drill piece. And as we got initial hole in, we'd change over to like a 6-foot long auger. Sometimes the drilling would get really tough because you might hit some rock or a root of the tree, but you just have to keep going and keep pushing that drill. And then once you got down, if it's a big stump, it'll be at least like a 6-foot hole, and it's about 2 inches across. We would then put down what we call a bully charge, where we take a short fuse, about 1 minute long, attach a detonator, and insert that into a stick of gelignite, and then light the fuse and drop it down the hole, and then push it down to the bottom with a long wooden pole. And we'd say, fire in the hole, and stand aside. And then a minute later, a bunch of dirt would be blown out of the hole, and you'd have this compacted cavity deep down under the stump. And then we would take a long fuse, maybe 6 minutes, which is sort of 6-7 feet long, attach a detonator and a stick of gelignite again, and then push that gently down to the bottom of the hole with the pole, and make sure the free end of the fuse is sticking out. And then we need to add more explosive, and that we'd use this ammonium nitrate fertilizer that'd been mixed with a diesel fuel. And then when we got the right amount there, we'd load the equipment back on the ute, and then we'd light the fuse and drive away, maybe half an hour. And then you'd wait, and then suddenly, boom.
Yeah, so exactly the sort of thing a young teenage boy wanted to do. We'd often be setting off several at once, actually. So it was quite. But one of the things you do learn from that experience, even at a young age, is to follow protocols closely. Maybe not quite as dramatic, but. So we've come back to working on VOC, CXCR4 recently, and this is really inevitable in some ways. There's been so much discovered about it in the last decade or so. It's overexpression in various autoimmune diseases, but also, and there it's a negative correlation where the B cells are doing bad things and a lot of B cells is not responding, is not going to help. But also in tumors, huge literature emerging that there's a positive correlation between CXCR4 in circulation or in the tumor and improved outcome, like numerous papers. Just to show you one, where this paper states that clonal tumor mutation burden and CXCR4 expression are the strongest predictors of checkpoint inhibitor therapy response. You're looking at checkpoint responder versus non-responder on this axis, and the upregulated in tumor reactive T cells on this axis. And CXCR4 is way out there in these T cells, correlated with response to checkpoint therapy. Now, T cell CXCR4 is suggested in these numerous studies to be acting by promoting tertiary lymphoid structure formation. But all of this data is correlation. And as we've heard or talked about, correlation is not causation. Now, the trouble is that as beautiful as the mouse is, and so important as a model for mechanistic studies of human disease, human conditions, there are a few differences in some cases. An important one in this case is that while CXCR4 is expressed similarly by stromal cells in mouse and human, it's not expressed in mouse T cells. So this expression in human T cells that is so robust and so correlated is not happening in the mouse. So what do we do? So what Fang-Lu Peng, a current postdoctoral fellow, is doing is taking a human bacterial artificial chromosome that has the CXCR4 gene in it and introducing that into mice to make transgenic mice, and including an iris amnion green reporter to facilitate tracking. And in one of the lines that he's following, we can see that compared to the control T cells in the mouse, these human back transgenic CD4 T cells, looking at the effector type cells, have expression of the amnion, and that correlates with expression of the protein inside the cell. The stromal expression is also there. As we said, it's a conserved feature, and this is tracking the human locus expression. So these are the stromal cells in the follicles, in the spleen, co-localizing nicely with, and it's the co-localized there with these complement receptors stained for the follicular dendritic cells over here. So we've got this model. So now we need to see if we can use it to see and test if the T cell expression is driving improved outcome, and if so, through what mechanism. This is very early days, so I've just got this one slide on where Fang-Lu is up to. He's looking at day 15, an intravenous transfer of a lung adenocarcinoma cell line model, so a lung tumor model. And we can see that the T cells in the lung, which include T follicular helper, T peripheral helper, and a couple of CD8 effector types, are highly expressing the amnion and the intracellular CXCR4, particularly the CD4 subsets. and that there is increased accumulation of B cells when the T cells can express the human CXCR13 compared to this control, and also of T cells accumulating, perhaps secondary to the B cells. So as I said, there's a lot to do. Fenglu's busy working away to test mechanism of what this CXCR13 expression of T cells is doing. So I want to come back to the recirculation concept now and to talk about why we were thinking about exiting after a few years. And so as I've mentioned, as we know, there's a lot was known in the mid-90s about how cells get into lymphoid tissues. And Steve Rosen's lab was one of the keys characterizing the importance of selectins in that biology. But at that time, really nothing was known about how they get out. And so that black box question was one that we began focusing on in the turn of the century, really, and took two-pronged approach to what we call the exit question. One was to follow this natural product that had just been characterized that seemed to be affecting recirculation in some poorly defined way. It was called FTY720. And the other was to follow up on early observations that infection and type 1 interferon could cause lymph node egress shutdown. Now, around this time, these signs were appearing around town. Exit, this is not a sign, it is a calling. You can see, might make some sense, but they could have been ads for the lab at the time. So we know lymphocytes are coming into lymph nodes and leaving at the equal rate in normal non-inflamed setting, right? And so entry and exit are matched. But what a group in Japan in this paper in 1998 reported is that when they treated with this natural product derivative that they had characterized in the context of immune suppressant screen they were doing, that it was inducing sequestration of circulating lymphocytes by what they thought was acceleration of homing. So they go in faster, then they're going to be less in the blood. But they did this in the rat, and they could cannulate the lymphatics in the rat, and they were observing also less cells in the lymph. So we interpreted that to possibly mean this was inhibiting egress. And so we said, well, can we do that in the mouse? So we got this synthesized locally at a huge expense, and we developed a technique to sample the cisterna chyli, the sort of collecting lymphatic vessel from the mesentery, for just a few microliters of lymph to avoid having to do live thoracic duct cannulations. This was a key step because we could then measure lymphocytes in the lymph at the time of euthanasia. And we indeed confirmed that egress appeared to be being inhibited. Now, while we were doing that, this molecule was being pulled forward by groups at Merck and Novartis as a potential immunosuppressant for use in humans, and they were starting clinical trials. And their pharmacology showed him it was getting phosphorylated in vivo. And the phosphate form, they realized looked a bit like sphingosine-1 phosphate and was acting on, or could act on, 4 of the 5 known sphingosine-1 phosphate receptors, which you can see are, again, this 7-transmembrane G-protein coupled receptor type. So given our observations here, we wondered whether any of these receptors might be acting in the lymphocyte for egress. And we noted that S1PR1, even though its old name was endothelial differentiation gene 1, is abundant in naive T and B lymphocytes. So we said, is it needed in the lymphocyte for egress? And we got Rick Proyer's S1PR1 knockout mouse that he'd been studying for other reasons. And Merdad, Charles, Lowe, Guy Cinnamon, Matt Lesniewski, Ying, all worked on this. And the key initial experiment, the knockout is embryonic lethal mid-gestation. So we had to make fetal liver chimeras. We take the hematopoietic stem cells, use fetal liver to transfer them into radiated hosts and let them reconstitute. And I want to thank Kevin Shannon for teaching us how to do fetal liver chimeras. And here's the first reconstitution, looking in the blood of the mice 8 weeks after constitution. The donor was a CD45.2 marked. So we know these are donor-derived hematopoietic cells, looking for the T cells. The wild type looked good, knockout, nothing there. So I remember my dad and I were like, oh, we don't really know how to make these fetal liver chimeras. We've screwed up. You know, we just couldn't believe that a phenotype, you know, as you know, those of you who do bench work, to get a strong phenotype like that is very rare. So we couldn't really believe it, but we said, well, we better check. So we found that there were T cells in the thymus. So we transferred thymocytes into the periphery and asked where they went. And we saw that we transferred these knockout cells in equal numbers into the blood, but they disappeared from the blood a day later. They appeared in all the lymphoid tissues, and they failed to come out in the lymph. So they could get in, but they could not get out. And so to summarize a whole bunch of work by a lot of people, quite a few people here, that we could establish that egress from the thymus and spleen into blood and from lymph nodes, Peyer's patches into lymph was dependent on S1PR1. S1P is high in the blood and the lymph and is made locally into these sites. And what Susan Schwab showed, who's here today from New York, is that the amounts in the lymphoid tissue are very low, and they're kept low by this enzyme called S1P lyase. And she showed that by following up on this interesting toxicology work on a caramel food colorant. And so if you want to know how a caramel food colorant led to that insight, you're going to have to talk to Susan at the reception over a caramel colored beer. And that food colorant is still in use, so you can ask about that too. So FTY720, Merdad and some of the other work we did showed that it's functionally antagonizing S1PR1. It's causing internalization of the receptor, so it can't function anymore in promoting egress. That molecule was FDA approved in 2010 for treatment of relapsing remitting multiple sclerosis. It's been put in by Novartis into over 300,000 people. Now, I highlight this to say that we were driven by a curiosity-driven science, as in how do lymphocytes get out of lymphoid tissues? We need to understand the biology. It was a curiosity-driven question. It led us to insights through this small molecule, which helped us tease apart the egress mechanism that helped Novartis get their drug approved. We were not working for Novartis. We didn't get any financial reward from Novartis. But they were able to cite our papers on mechanism, as in the mechanism, at least partly here, is that if you block cells getting out of lymph nodes, they won't be able to go to the site of autoimmune attack, in the case of multiple sclerosis, the brain, and you'll have immune suppression, you'll have protection. So they could cite that, and that helped them get approval for the drug at the FDA. So doing curiosity-driven science enables discoveries that achieve translation and has important value in and of itself. Now, I want to come back to the second prong we were taking to the egress question. And that was the shutdown process. And this is where an infection will cause, the cells are coming in and out at equal rate, but if you cause a local infection, look locally from that lymph node, egress into that efferent lymphatic is shut down. And that was actually first shown in the sheep, where the efferent lymphatics are larger, but that was not just in the sheep, it was in Australia in the 60s. And this allows me to sort of connect to the livestock theme of the talk, is that, I don't know how many of you know, but the biggest export from Australia from the late 1800s up to about 1960 was wool. There were sheep everywhere. I mean, these flocks were 100,000 or something, I don't know. So getting a few sheep with efferent cannulations was not a problem. And I noted in the paper, they said they used merino ewes and wethers, which, merino was the key breed because its wool is the best. But of course, that industry has shifted to synthetics and such. So interferon is a key downstream mediator here in the shutdown. And so we took advantage of the interferon to try and understand what was going on. And what Merdad observed early on is that S1PR1 surface expression was reduced by interferon exposure, but the mRNA was not, looking like some translational process. Because we came to thinking, well, the shutdown could involve some regulation of S1PR1, given by this point we'd implicate S1PR1 in being needed for egress. And so that looked plausible. But we also knew that interferon is a huge number of genes it induces in responding cells, one of which is CD69, an immunologist's favorite activation marker. Now, around that same time, two groups published papers showing that when you transgenically overexpress CD69, it represses thymic egress. And so we put these things together and came up with a model, which then Larry and Merdad tested, that I'm just going to summarize here, that interferon signaling induces many genes, including CD69, which is a type 2 transmembrane protein, dimer, that we showed physically interacts with and inhibits the function, egress promoting function of S1PR1. And that will also include internalization and destruction of the receptor. Now, in addition to innate stimuli and interferon, we and others knew, of course, that antigen receptors can induce CD69, and that will contribute, when you have a recognition event, to transient retention of the responding cell in the responding lymphoid tissue. And then in the years since, it's been learned that transcriptional reprogramming to induce constitutively CD69 is a part of many kinds of resident memory to keep cells in a peripheral tissue. But we still didn't fully know from this work how CD69 was inhibiting S1PR1. Now, Alex Bankovich, who's here today, did some beautiful domain swapping work and mapped the interaction of CD69 through the transmembrane part with the fourth transmembrane region of S1PR1. But it was it's still hard to work out from there exactly how it was working. And so we were fortunate just 3 years ago to collaborate with Xiaoshun Li at UT Southwestern, Hongwen Chen's lab, expressed the complex purified it, did cryo-EM, got a structure, and found this, indeed, there was this large 600 angstrom interaction surface between CD69 and S1PL1. And that involved, Alex was happy, transmembrane 4 of S1PL1. But very important here is that it was putting the receptor into an active conformation with G-alpha-I bound, as you can see in the structure there. So it is effectively a ligand, a cis agonist of S1PL1. So when CD69 is turned on, the receptor will no longer sense S1P and there will be no ability to egress. So it explains how it can act as a shutdown mechanism. So moving on to the third segment here of the science, and that is the real-time imaging. And so we launched this initially to see if chemotaxis was involved in follicular exclusion. And because I had a fortunate interaction with Mike Cahalan, who introduced two-photon microscopy to immunology. And Taka, who's here from Japan, really made this work happen in the lab through initially a collaboration with Mark Miller, and Taka would travel down to UC Irvine, where Mike Cahalan's lab is, and have cells that were flown down, and they would coordinate the transfer and the imaging and get some initial data. It was all amazing. The two-photon microscopy, just to back up, allows one to see deep into a living tissue, some couple of hundred microns, and look at fluorescently labeled cells in a live state. And so that we launched that way. But then once we're into that a bit, Max Krummel we teamed up with, and he went ahead and amazingly built a custom two-photon microscope himself here at UCSF. And so we really benefited from that, and I really thank Max for that. And so looking then into a follicle where we've got antigen engaged B cells, the antigen was just injected when we started the imaging in green, and non-specific B cells in red. This is the follicle. The T-zone is sort of deep down here. We're looking through the capsule, and hopefully you'll see, all these movies are accelerated somewhat, but you'll see as the time-lapse movie plays that the cells are accumulating over here, that they've moved into this excluded location. And we were able to relate that to the distribution of CCL21, this T-zone chemokine, and see that it spread about 200 microns into the follicle. So it could support this, what we showed by tracking was directed movement. So real chemotaxis in vivo, one of the first sort of demonstrations of definitive chemotaxis in a lymphoid setting. So coming to the dynamics then, sort of moving from that initial imaging work that Taka did to ask further questions about looking at the dynamics of the B cell response. We, just to sort of back up, we know that lymphocytes are coming in from the bloodstream all the time into a lymphoid tissue to survey for antigen, very rare specific cells. And the antigen is going to come in by the afferent lymph for a lymph node. There's a Tree Fan, who I don't know if he made it back, he was here recently from Australia. When he was in the lab, one of the things he did was image that process. So here we're looking into the follicle through the capsule. The blue is the side of the capsule, and here's a lymph-filled sinus, and this is the follicle. And the antigen is in red, and this is in minutes and seconds after vaccination or injection. And so you can see within a few minutes after you get that jab of protein antigen, it will be filtering into your lymph node in the subcapsular sinus. And some of it will decorate cells over the follicle, allowing B cells to have some initial chance of encounter. Now that process is happening fast, but the antigen can't stay there for too long. It turns out that the follicular dendritic cells that we mentioned are able to hold antigen on the surface for long periods in a stable form. And so that allows B cells to survey for longer periods. And this is something that Kazu asked, can we see B cells capturing antigen from the surface of follicular dendritic cells? And Kazu is here from Japan today. And so the antigen now is red again. It's on the follicular dendritic cell inside the follicle. And we've just got a couple of B cells in green that we know are specific for that red antigen. And so we can see that not only are they surveying, but they're physically capturing the antigen and taking it with them. And that is then going to allow them to internalize, process, and present some of that antigen to helper T cells. A fundamental concept, of course, of how B cell responses work in getting T cell help. And so that antigen engagement, we're moving up over here, is increased CCR7, as we saw earlier on, shifting the cell to the T zone. And this is a foreign antigen. So there's MHC peptide being presented. It's allowing the helper T cell then to lock onto that B cell and provide help. And that was something that Taka imaged, again, using two-photon microscopy, where the the cognate B cells in red and the T cells in green. You can see these beautiful conjugates form and can last for the full movie there of an hour in some cases, where signals are being exchanged from the helper T cell to the responding B cell. Now, what's harder to capture, because these can last a while, is the actual encounter events. But occasionally you can do it, and Taka did it sometimes. And so I'm going to show you one example and see if you notice anything familiar about that. No sense of familiarity yet. I'll play it again. Can you see that? Can you see that? Can you see it now? You know, it took us this event and like 30 years to make our first lab T-shirt. But you know, it's a lot of fun to sort of be celebrating not only interaction of these cells, but to have everyone together to celebrate as well. So coming from that helper stage, some of the cells will go on to become antibody secreting cells quickly, early antibody protection, but some of them will go on to form germinal centers, which, as some of you know well, are sites of antibody affinity maturation by iterative cycles of proliferation, Ig gene somatic hypermutation, and positive selection of rare gain of fitness mutants. So low affinity cells come in, and high affinity cells some days or even weeks later are coming out and giving rise to antibody secreting cells and improved protection against the antigen. And that Darwinian process is taking place inside the follicles over the follicular dendritic cell network, where the antigen is available for testing the cells out. And there can be many germinal centers forming across many follicles. And so if you look in a lymph node section by just H&E, classic staining method, you'll see these germinal centers in a responding lymph node, or just one zoomed in over here. In fact, they're so evident that they were observed first and named in late 1800s. And this is a hand-drawn version. Remarkable how similar they look. But remember, that's a fixed tissue. If you actually image and look at what's going on, as Taka and Chris Allen did, where they've labeled about 5% of the germinal center cells green with GFP, you can see that they're actively exploring the environment for antigen, for undergoing cell division, cell death as part of these selection processes. So one of the questions we became focused on was, how does this confinement occur? With the quest then of trying to understand why. And so just to summarize a bunch of work in one slide about how, what we observed, actually going back to Chris Allen's microarray data, so tells you it was old, that S1PR2 was highly expressed, this G protein coupled receptor, selectively in germinal center B cells. And this one, unlike S1PR1, the egress promoting receptor that couples to G-alpha-I, this one couples to G-alpha-13 and inhibits migration. And work by particularly Susan Schwab showed that S1P is present in a decaying gradient from outer to center follicle. So if cells with S1PR2 try to move out, they're going to be inhibited in the migration, they'll be confined. And so when we look at S1PR2 deficient mice, as Jesse Green did when he was in the lab, we see a partial deconfinement effect for the germinal center. Now, when we looked in the G-alpha-13 deficient, this downstream component, we see a more severe deconfinement. And this led us to think there might be another receptor upstream. And this was something that Juggin Rapidi looked at and found that there was this second receptor, P208, that could act in a similar way through G-alpha-13 to promote confinement. And so we want to understand more about that receptor. And so Eric Liu deorphanized it through classical biochemistry and identified S-geranyl L-glutathione, or GGG, shown here, as a nanomolar potent ligand. And I just have to say that I'm really proud that Eric purified that from a liter of pig bile. And Eric and Antonia and Finn showed that GGG is also in a decaying gradient, or a differential between the outer and the center follicle. So the logic makes sense. So then we've got this understanding of how the confinement occurs, so we can start to ask why. And this is something that Kevin Chen's been doing. He actually just graduated last week. Is to test the hypothesis that germinal center confinement promotes antibody response diversity. So if you think about the evolution, the Darwinian evolution occurring in these separate confined structures, you can imagine even in response to the same antigen, you might have a different evolutionary outcome, and that there might be different specificities to the different parts of the antigen winning out if you have separate germinal centers. But if you have this deconfined setting, there might be these early arising winners that just sweep the landscape and diminish the diversity. And keep in mind that if you look in a macaque, as Darryl Irvine did, or in a human lymph node, you're going to have something like 100 germinal centers. And so a lot of opportunity for diversity in this way. And simple-mindedly, you could think about it like the Galapagos Islands and Darwin's finches, which could not fly between the islands and evolve these different beak shapes. I mean, we're thinking about different antibody shapes. So to show you just two pieces of data from Kevin's work. First of all, can we see increased, the deconfinement, this perturbation allows us to increase exchange between germinal centers. So on the left, the germinal center B cells are labeled in orange and they're nicely confined, but there's a few outside, and Kevin tracks those few, and you can see they're moving around. But he wasn't, in these admittedly short time frames of 45 minutes, able to capture any that actually exchanged. But if we look in this perturbation, the deconfined setting, you can already see that there is more cells outside. There are still structures, which is necessary for the model to be tested, but there's more orange cells outside. And then when in this 45 minutes, when Kevin tracked in those yellow tracks, you can see occasional cells that he could observe passing from one germinal center structure to another. In additional methodologies, particularly spatial sequencing, he established increased clonal sharing between the deconfined germinal centers. So then, coming back to the model then, if we think about a typical complex antigen, it will have dominant determinants and subdominant ones. And so keeping a lot of diverse, normal confined germinal centers might allow some germinal centers to have a good response against a subdominant and not be taken over by these more readily responding dominants. But if you have a deconfined setting, the dominant cells may just take over the landscape. And so the antigen we use to explore this, it has this hapten as the most accessible and thus dominant epitope, and this core protein of albumin as the less accessible subdominant. And so when you immunize with that, compared to the normal situation, in the deconfined situation, the fraction of germinal center B cells receiving the dominant epitope is increased and the subdominant is decreased. And if you look at the affinity of these dominant responders, they are also somewhat higher. So the data provides support for the model then that germinal center confinement promotes antibody response breadth. So finishing up the science and then lots of thank yous. I want to take us through the sense for a B cell response. And this is a partial list, and there's quite a few people in the audience who've worked on other cues that are playing important roles that I haven't had a chance to mention. But we know that lymphocytes are continually coming in from the blood, and the B cells got their CXCR5, and they're going to be attracted by the fragrant FDC into the follicle as an explorer B cell. And they're going to spend time exploring around. But if they don't find any antigen, they're going to get bored, and they're going to turn up S1P1 and exit and go to another lymphoid organ to look again. But if they do see antigen, they're going to become excited and relocate in response to the scent of the T zone with their CCR7. But if it's self-antigen, they're going to be, these are narcissistic cells. They're going to die. But if it's foreign antigen, then they become engaged B cells with T cells, get help to undergo clonal expansion, give rise to progeny B cells, some of which rapidly become secretor B cells, make a lot of antibody, but some become mutator B cells, go back into the follicle, and they turn up this S1PR2 and P208 and suddenly become very aware of the foul odor around the perimeter of the follicle. And so they stay confined where the antigen is in the follicle center, the fragrance of the CXL13. Get help there, mutate, select, repeat that a number of times, and some of them can come out as selected high-affinity long-lived plasma cells or secretor cells and memory cells. And if this is happening across 100 islands, we get greater overall diversity. So with that, I'm going to say that there's still, of course, much more to do. I'm not going to dwell on these points. There's a lot of these things are going on in the lab. It's really to say that we're not retiring, that we've got a lot to do, and we're still doing it. And really, in the remaining few minutes, to say a number of thank yous. First, to thank so many people across UCSF, collaborators, colleagues who've contributed in so many ways to our work, to everyone's work in the lab over the years, and just it just makes this community such a great place to be, and just to want to stay and keep going. And of course, funders, I've been very fortunate and thank really generous funders. And then, as Mark mentioned earlier, the biomedical sciences graduate program has been so important to the immunology community, certainly to my lab, bringing in fantastic students and supporting them. And as Mark mentioned, I was director for some time. This is from one of our Tahoe retreats. And I think some of you in the audience might be able to see yourself, your younger self in there. But I really want to use this to thank Lisa McGargle, who was the program coordinator while I was director, and was just phenomenal in supporting the program, keeping it humming, paying attention to detail, paying attention to student concerns, keeping me up to speed, but also seeing the big picture and really caring that education of students and the training process is so important for our success, but also for the nation. And so I really thank Lisa for that. I want to thank my colleagues in the department, just such a great group to be part of this community. And maybe even more importantly, to thank the administrative group behind us who really work our way so much to keep everything functioning and making it possible to do what we do. And so I particularly want to highlight Claire, Kang, Manlio, Annie, Sammy, Erica, and Bernarda. But really so many people in the department support our work. And then Jonathan, who's really in ImmunoX, for just endlessly supporting immunology program and my efforts with the training program over the years. So thank you all. And then I've got a few photos from the lab over the years. I'm not going to say out your names. Many of you are here, know who you are, but the year, for your curiosity, is roughly, I couldn't quite write down the bottom there. And so thank you so much and for so many of you coming. This is when we had a resident artist in the lab. This one over in Angel Island, you'll notice that we have an AT shirt on here. And if you look closely, it says it's been rebranded Corona Extra. And even the beer, we take stuff seriously, right? So if you want to know what that's about, you're going to have to ask Larry at the reception. And then when we had AI, I think here in San Francisco back in 2011, we had this little get together in our backyard. And I think almost everyone who's in this photo is here today. And you can see Tanya back there taking care of the livestock. Up in Golden Gate Park, here in our beloved courtyard. And then a couple of years ago, we had a sort of Bay Area get together of alumni, really to thank Jinping and Ying. Collectively, I think it's 45 years of support for the lab. And as research associates, they're just so skilled and make so much work. Things happen to keep the lab working and supporting everyone so diligently. And so I and I'm sure all the past lab members here are so grateful to your work. And so as we've heard, there's people from around the world here today. This is the alumni global diaspora. I'm just so appreciative and just so thankful for the work that we've done together and being able to celebrate here today. And thanks so many of you for coming. You'll notice that with some people have come a long way, but there are a lot of people who seem to like the West Coast here. And then finally, for the lab, so I thanks current lab members that we've still got a lot we're doing. This photo is actually already a couple of years out of date. So we got one just last week where we're doing the hooray for antibodies pose. And then finally, to thank my family, there's my brother, Mark, another Mark. Thank him for teaching me so much and keeping me on my toes. And my parents for connecting me to biology, just from so young, for teaching me an honest day's work, and for being so supportive. And then finally, my family for bringing so much joy into my life. So thank you.
We have for you.
Thank you.
questions, and I think we'll accept them from Zoom as well. So,
Questions? Mark.
Well, Jason, I was going to start with a question about something that
Everybody is talking about in the autoimmune. I was going to start with a question related to something everybody's talking about in the autoimmune disease field right now, when going in and blowing up B cells with either CAR T cells or bispecific engagers. And I'd love to get your thoughts on what are we actually doing to unwind the autoimmune response?
What are they doing as in moving them?
Right, where you're getting these patients with lupus, they're going into remission.
Right.
Right. What are we doing inside the scent of the follicle?
Yeah, well, I think the beauty of these engineered systems, whether it's the CAR T cells or these bispecific antibodies, is just the depth of the clearance of the B cells is more complete than previous approaches, right? And so some of the past B cell depletion therapies weren't getting into lymphoid tissues as much as they could clear systemic B cells. So while it looked good in the blood, there were still pockets of cells, much more than probably realized, in lymphoid compartments, which you can't readily sample in humans. So these new technologies are depleting much more deeply. But yeah, how is achieving this potentially long remission even after B cells come back? There's a lot we need to understand about whether the resetting of the repertoire. One possibility is that Chris Goodnow, for example, has contributed to, is that there's been somatic mutation of proto-oncogenes in some of these autoreactive lineages that contributed to that disease development. And so if you wipe that out and reset, then even if there's a predisposition for that, it's going to take a long time to get back to that sort of setting point of that type of autoimmune disease. I think that's just one version. There's going to be others, and impacts on lymphoid tissue that relate when you move the B cells. Is the effect that maybe you're sort of getting at on the lymphoid tissue more related to what I've talked about, and lymphotoxin having secondary effects that play out on the T cell compartment even after the B cells come back, that there's been a reset there as well. I think these are also possibilities. I talk too long, right? Here's one.
Okay, this is a more basic question, but I feel like the story alluded to all of it. But yeah, what are the main reasons for egress shutdown upon infection? Is it just to maximize the surface contact area between naives and the antigen,
right, great,
or for that optimization journey that happens after activation and lock them in?
Right. I didn't explain why shutdown is thought to be important. I think we probably still haven't, the field hasn't done all the mechanistic tests of it, but the idea is that by shutting down, but still having entry going on, and we've been studying Kevin Chender's nice work on how the entry continues to go on in the inflamed node, the lymph node will enlarge so that the cells are staying there longer and not wasting time going off to other lymphoid tissues that are not draining a site of infection. So you increase the number of cells, even if it's still only 1 in a million. If you now have 10 million instead of 1 million cells, you've got 10 instead of 1 cell that could see the antigen. So that simple logic is how we think about it. I think there's still tests that can be done to further explore it. Yeah.
Hi, I was wondering, within the follicle, is there physical exclusion between the quote explorer B cells and the mutator cells? Have you looked into that?
Right, that we've got the confinement of the mutator B cells, and another reason they're confined probably is because they're so dangerous as mutator cells, and germinal lymphomas are a common problem, and if they disseminate, then they're a real problem. So keeping them confined while they're mutating is another reason. But are the naive cells actively kept out? Is that maybe the question? And we are studying that. We think they are. We don't fully understand how.
I guess so. I'm relatively new to B cell follicle biology, but there's the naive cells that go into the follicle, as well as the mutator cells that are in the follicle. And are they encountering each other in their different states?
Are they encountering each other? They are, as you say, right next to each other. The mutator cells are antigen specific. That's how they've ended up as mutated. They're really dividing and responding. The surrounding cells are still polyclonal. So they're very rare cells that are specific. The few that are specific can be drawn into a response at any time if they get the antigen. So where they can go into a germinal center and do that, there is some evidence for that happening at some level, yes. But they might also see it in the more conventional way of going through the same steps that I showed you, that they'll be excluded to the T zone, interact there, and then potentially go into that ongoing germinal center. And the B cell memory response can involve that kind of process.
Jason, just to follow up on your response to Mark's question, have to ask a cancer question, of course. You mentioned a lot about egress in and out of lymphoid organs, but you also mentioned that with these therapies, you see a lot more cleaner loss of those B cells in the periphery than in the lymphoid organs. And so it raises a question of what sort of survival factors are going on in these lymphoid organs and how might they be contributing to autoimmune disease?
Yeah, thanks, Kevin. I think that these ectopic sites are turning on a very similar set of factors, but I shouldn't say I fully know. I think BAFF is made by this B cell trophic factor by many cell types, stromal and hematopoietic. And so once you start forming a structure, local BAFF is being generated and likely contributes. Whether some other cytokines may be as well, but BAFF is so important for B cell support that I would think that that's what's getting enabled in those niches. But yeah, the factors determining its availability are still, I think, not well enough understood and should be studied more. Right up the back. And
Great talk. So I was wondering, how far does the scent travel? So you have talked about signals within the lymphoid tissues, but what's the frontier of that scent? Is at the endothelial level, recruiting the lymphocytes? Do these signals, or there may be some signals that travel farther away, trying to recruit these lymphocytes to the lymphoid tissue?
I think each type of signal has a different distance over which it can act, but the chemokines, when they're expressed in the tissue, and then to have a boundary where they're not expressed, over what distance does the effect play out? From Tacker's work that I showed very briefly, we measured about 200 microns of signal we could detect, and it mapped quite well to where we saw the directed movement begin. So there was a harmony there that made a lot of sense. And then Michael Sixt, who's a leader in looking at some of these things, has found a very similar distance for CCL21 in the skin, pulling in dendritic cells. So there may be some biophysical logic to that distance for some of these types of molecules, but I think some other molecules have much shorter distances. I think sphingosine-1 phosphates metabolize so fast that we don't actually see for S1P1 any evidence of directed movement. It's really a decision point at the egress site that Irina Grigorova showed years ago. So yeah, differences depending on the cue, and that may be also why there's these very different types of cues. They allow, among other things, different shapes to these gradients.
Yes, a quick question. So once the so once the reaction is complete, you have this big lymph node now. What controls the time at which it takes before like S1P gets re-expressed, or before they can start to exit again? Is there
the exit process, even just for the normal transit?
No, well, transit, yeah, but after, or it gets if you block it formally, and then also in the
so the activated cells. For the T cells, we did more, I think. Trung Pham, who's here from Stanford, showed that it takes about 3 or 4 divisions. So a T cell TCR signaling not only induce CD69, it will downregulate the transcription of S1PR1. So there's sort of a double block in egress. The CD69 works faster and the transcript takes a bit more time to decay. But after about 3, I think, or maybe 4 divisions, Trung saw S1PR1 come back up. And when you look in distant lymphoid tissues, it's about that division number that you start to see cells appearing. So there's some internal logic to that, yeah.
Howdy, thanks much, Jason. Jason, this is about cells that are retaining lymph nodes. It's going to be a T cell question. My question is, do you see a role for this kind of working on signals to move? These are HIV reservoir cells, what I'm talking about. HIV infected cells that are sitting in lymph nodes that are inert, that are sort of hidden from therapy. Finding a way in this kind of approach to push them out to promote egress?
Yeah, certainly people have thought about that, and I don't know if Rachel Rudishauser is around, but that the T follicular helper cells were thought to be a reservoir. And in fact, one of the early studies on HIV, it was done in Perth actually, before it was even called HIV. I think they noticed virions in these immune deficient patients on the FDC. And that was supported later by famous people. Yeah, it's a funny story because my brother was dating the daughter of the physician who had that paper with these things. That's why. So I think this reservoir logic has been thought about a lot. I'm not expert in it, even though it's really important, as you indicate. I think it hasn't held up that the reservoir is that restricted to that location, that it's not that TFH restricted. And Rachel can tell us later if we see, or maybe others know as well. And so it's not clear that just pushing things out of the compartment will be an improved way of depleting the reservoir. But I think we should keep it in the list of things to be thinking about. Yeah.
We have a Zoom question. Are human germinal centers bigger than mouse? And what about elephants? And that's from Christopher Goodnow, who you mentioned.
Yeah, I didn't hear the last part.
It was good. It was Chris.
Oh, that was Chris? Chris, you haven't read the latest preprints. There's one out showing that they measure the germinal center sizes across the whole series of species. And they might have had the elephant in there, but they found the sizes were all the same. So that, I think, could be argued to fit with the sort of Darwinian logic that it's already optimized, doesn't matter what the species is, to achieve an appropriate amount of selection and diversification. And then you add more diversity by having a larger lymphoid tissue with more responding germinal centers. But there's some real experts here later too that'll debate this with me, and maybe Chris can do it online too. But thanks for joining us. Yeah.
With that, we're at time. So yeah, thank you so much.